Hello guys, welcome back to my channel. So I'm Himani Agarwal, and today in this video I'll be continuing the topic which I was discussing with you guys, and today we'll be seeing the process in detail. If you're not seen the previous video, the link is given in the description. Please go and check it out. So let's start with this process today. And before coming to the process directly, I want you to know some important topics regarding this. So I have discussed about the promoter region, right? In the previous video, I have discussed that the promoter region is the first region which the transcription apparatus recognizes and then binds to it. So there are some important points which you should know about a promoter region. Every living organism has promoter region for their transcription process, and this promoter region has different kind of nucleotides. But apart from the different kind of nucleotides, it has got some similar kind of nucleotides which the promoter region has in each and every living organism for a particular species. Okay, these are called as the consensus sequence. CONSENSUS, consensus sequence. Okay, this is very important and you should know this term. It will be used many times in this topic. So, consensus sequence, what is consensus sequence? Consensus sequence is a sequence which is similar and has similar kind of nucleotides. In case of prokaryotes, there are two important consensus sequences which are called as -35 consensus sequence and -10 consensus sequence. Their names are given based on the origin where they are located. -35 consensus sequence is upstream to start point of the transcription, and it is upstream -35. Similarly, -10 consensus sequence is upstream to the transcription start point, which is 10 nucleotides away from the start point. -10 consensus sequence is referred to as TTGACA consensus sequence. But a very important consensus sequence is the -10 consensus sequence. This is also called as the Tata box, TATA box, or the Pribnow box, PRIB, Pribnow box. These are the two consensus sequences which the transcription apparatus recognizes and then binds to. The transcription apparatus which I have discussed the previous class, it is the RNA polymerase. So RNA polymerase will be binding like this. Whole sequence will be under the RNA polymerase enzyme. Okay. So these are the important points which I wanted to discuss before coming to the process directly. So coming to the first step of this process is the initiation. So what is initiation? Initiation means to start something, right? Which leads to a process. So when RNA polymerase recognizes the -35 and -10 consensus sequence, it binds to it along with its sigma factor. Sigma is denoted by the sign like this. It is a Greek term, sigma factor. In the previous video, I have discussed about it. Please refer to that. After binding with the sigma factor, the RNA polymerase is encloses to the DNA. Okay, and then it forms a transcription bubble. Can you see this term, transcription bubble? What is this bubble? The double DNA helix is opened up for the RNA molecule to be formed. This region is the DNA-RNA hybrid. Okay, I can change the color for your convenience. So this is the DNA RNA hybrid. In the last video, I have said why the RNA molecule is growing. The first nucleotide which is added is a triple PPP, triphosphate, because it does not participate in any kind of phosphodiester bond. But the second RNTP which is added, it forms in the phosphodiester bond, and therefore it has only got one phosphate. Remember? Yeah. So this grows and grows and grows by addition of RNTPs, which is the raw material, which is the substrate for transcription. Okay. So, but in the initiation process, during the initiation process, the RNTPs which are added, RNTPs which are added, they are not more than 10 RNTPs. Okay, that's why only short transcripts, short mRNAs are released into the cytoplasm. This is only the starting, a start of the process. Okay, as soon as this RNA polymerase crosses the consensus sequence and enters into +1 region of the DNA, which is the actual starting point of transcription, a long RNA molecule greater than 10 nucleotides are added. And then it is said that this RNA molecule has accomplished a ternary stable complex, where the RNA polymerase, the DNA, and the growing RNA chain, they are in stable conformation. It can proceed towards the elongation process. Now coming to the elongation process, what happens is that this hydroxyl molecule, which has already been attached to the growing RNA chain, attacks the incoming alpha phosphate to form the phosphodiester bond. What happens during this process? The extra two phosphates which are there, they are released in the form of energy. 2pi are released in the form of energy along with one water molecule. Okay, this is what happens. So when the next RNTP will be coming, this hydroxyl molecule will be attacking the next phosphate, and this continues. This is the elongation process as whole. Coming to termination, right? Termination means what? It is the end of the process. This is where DNA says, no, enough. I don't want any of the information to be transcribed further. You can stop now. RNAP, your job is done. You can stop now. But this process also occurs via two ways. It can be a row-dependent termination, or it can be a row-independent termination. You must be thinking, what is exactly this row, right? Row is written this way. It is a kind of P. In English, how we write P, it's like this. It is in Greek. Even this is in Greek. It is a protein molecule which helps the process in termination. Okay, so there are row dependent termination means what? The termination is dependent on this factor. The second is row independent termination. The termination is not dependent on row factor. First, coming to the row dependent termination. So for this, the RNA molecule which is growing, it does not have any secondary structures. What do you mean by secondary structures? In RNA, there can be something hairpin kind of structures, due to which it binds to itself. The bases are complementary to each other and they bind to themselves, and therefore there leads to some secondary structures. Okay, so For this kind of termination to occur, the growing mRNA molecule should not have any kind of secondary structure. So when Rho recognizes such kind of mRNA, it comes and binds here. This is the Rho molecule. This is the Rho factor. It binds to the growing mRNA chain. And then second thing which is required is in DNA there is some kind of termination sequence. I'll repeat this. In DNA there is some kind of termination sequence. Okay, termination sequence, due to which RNA polymerase, which is reading the DNA sequence in downstream, it will halt, it will stop, it will pause for a moment, due to which this Rho factor will pace its movement and it will come here. This Rho factor has an helicase activity. So, due to the helicase activity, the mRNA molecule, the growing mRNA molecule will separate from the DNA RNA hybrid and along with the Rho factor. So after this we will get mRNA plus Rho factor. It will disintegrate from the mRNA. Now this mRNA will go directly into translation. Translation I'll be discussing in my next upcoming videos. Okay. Now let me tell you something about Rho independent termination. We have seen what happens with Rho. Now we should know what happens without Rho. So for Rho to happen, there are two necessary things which should be there in the DNA molecule. Okay. First are repeated inverted sequences. Repeated inverted sequences. Okay. So what happens if you have an inverted repeated sequence? See, I'll give you one example. A, G, G, T. Okay. Here you have, what did I say? Repeated inverted sequence. Here you will have T, G, G, A. So suppose this is transcribed. These two sequences are transcribed. What will you get? U, C, A. And here what will you get? This is for the first sequence, right? Here what will you get? A, C, tell me, U. Right? Am I right? Yeah. Okay. Let's see it once again. What is inverted, repeated inverted sequence. So it will be T, T, A, A, G. So it will be repeated inverted sequence. So it should go this way, but the sequence should be inverted. So G inverse is C, A inverse is T, T, A, A. Now, when this is transcribed, what will you get? A, A, U, because it's RNA, U, U, C. Here you will get G, A, correct, A, U, U. Yeah. See, now what happens because of this sequence, they will bind to each other. They are complementary to each other. So A is complementary to U. This A is complementary to this U. U, let me change color for your convenience. This U is complementary to this A. This U is complementary to this A. C, G. You can see this? Because of this, RNA will bind among itself. A hairpin kind of structure will be formed, which girls use for tying their hair. That is a hairpin structure for RNA. All right. Yeah. So you can see this. This is formed. Second thing which we require for row independent termination is a sequence in DNA, a long polynucleotide sequence, A, 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 A. When this is transcribed, what will you get? U, 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 U. Yeah? Because of these two things, transcription will terminate. Why? Because of this. Because AU bonding is weaker than GC bonding. So it is easier for the DNA RNA hybrid. It is easier for the RNA from DNA RNA hybrid to terminate itself, to come out from that hybrid and be independent in nature. So this mRNA will now come outside of the transcription apparatus and DNA will reconcile itself. And RNA polymerase will also dissociate for the next transcription process to take place. Okay, so guys, this is all about transcription process. I think I have explained it in quite detail, and I've started from very basics till the end. Thank you very much. So please like, share and subscribe to my YouTube channel, and keep looking for my next videos. Thank you very much. Stay safe, stay happy. Thank you.